My name is Miss Hennigan. I'm a biology teacher at Taunton High, and with me is Mrs. Sanborn, who also teaches biology at Taunton High. Because of the remote learning, we've forced to do some of our laboratory activities remotely like this. So keep in mind, if you are doing some labs, you may have a different teacher showing you a lab that we all us biology teachers do. But and maybe your biology teacher is Mrs. Shaw or Mrs. Carrera, but you're seeing the same lab that she would do, but you're doing it with us. So this morning we're going to talk about using the scientific method to identify unknown samples of liquids. The first thing we need to talk about, though, is our safety tools. You notice that Mrs. Sanborn and I both have our safety goggles on, our gloves, our hair is tied back, so that we're following all the safety rules that we need to be involved with today. The other thing that we're going to do is when we actually check these samples for odor, we're going to use the wafting technique. Now you should have seen these things already in the precursor to this laboratory, and you should also have gone through the steps of the scientific method, and you should have an overview of what pH is already. There's going to be a go-formative lab report that you'll be able to fill out, but for now, let's sit back and watch what happens in the laboratory. So the first, notice that all the samples are labeled 1 through 6. I'm going to test sample number 1. First of all, you notice that it has sort of a yellow color, and it's very liquidy like water, okay? It's not thick at all. So the first thing I'm going to do is I'm going to carefully, I only open one sample at a time. I'm going to open it up and I'm going to waft it. This smells very sweet and fruity. Mrs. Sanborn and I already know what's in these samples, but you don't. So be very careful when you're doing your wafting. The next thing I'm going to do is take a little piece of pH paper, looks like this, and we're going to measure the pH of each sample. So let me notice I take one piece of paper out at a time. It looks like this. Then I'm going to dunk it into the sample. I'm going to put the cap back on the sample. And I'm going to lay the pH paper right on the top of the cap. Now, I take and I use my container, which is numbered from 1 to 11, and I'm going to measure the pH of this sample. And the pH of this sample is approximately 3. Okay? So 3 to 5, somewhere in there. All right? So that's sample number 1. You should be recording this information. What I said about the color and the viscosity, as well as the odor, as well as the pH.
Miss Hennigan, what does viscosity mean?
That's a good question, Mrs. Sanborn. Viscosity is the thickness of a solution, or the thinness, if you will. How thick a solution is. Now you remind me, we need to talk about the most important safety tool too before we finish this. But, okay?
All right, so I'm going to do number 2. Number 2 is, when we look at it, it's kind of white and opaque, which means you can't see through it. It's very thick. It's actually almost like a milkshake consistency if you look at it. And so I'm going to take the lid off and I'm going to waft it. And it's kind of minty, lightly minty, okay? And so I'm going to take a pH paper. They kind of stick together sometimes. Return the rest to the container so I don't get them contaminated. And then I'm gonna dip it in. Now it's got some white stuff on it that was some of the material. It's kind of obviously kind of sticky. And
big difference in the color there, though, Mrs.
oh,
Sanborn.
yes, very much so. Completely different part of the pH scale. So if you look, I got the numbers upside down. If you look, it's about 9, almost perfectly, okay? So this end of the pH scale is acidic and this end is basic, and neutral's right here in the middle. And so it's kind of towards the basic end, right? So,
Great. Now we'll do sample number 3. Sample number 3 is very liquidy, not viscous at all, clear solution, and yeah, that's good. I'm gonna waft it now. Whoa, this one you need to be very careful of. We know what it is, but this will burn like your little nostrils out, so you definitely want to be careful when you're wafting this number 3, right? So
I can smell it all the way over here.
You can smell it over there, right?
Yeah.
Exactly. Get a strip of pH
Smells
paper.
kind of like cat pee.
Cat pee. That's a good, there's probably some of it in there.
There is.
Okay, so I'm going to take a pH paper, I'm going to dip it in. Whoa, look at that color change. Now, when we look at this one, we're going to see that, again, this is extremely basic. It's the highest one it will go. It's almost black. Okay, so this has a very basic pH at number 11.
All right. So I'm going to be doing number 4. And 4 is actually very liquidy. It's a pale yellow, and it's not really completely opaque, maybe translucent, which means that light can kind of get through it and stuff. It's not like flat completely. There's kind of, you can kind of see a little bit through it, but not very well. So I'm going to take the lid off and waft. Okay, so it smells sour but fruity. Okay, sour and fruity. I like the smell.
Yeah,
Yeah.
it's a good smell.
Yeah, so I'm going to remove a pH strip. And we'll do our little dip. These lids don't like to go back on. There we go. And let's see if I got it facing in the right direction. I do not. So it's probably at this end somewhere. It's probably between 1 and 3 somewhere. It even looks darker, but I think over time this has turned a little bit pinker than it actually would be.
Yeah.
So it's definitely in this area. So it's definitely in the this end of the pH scale.
Super. Okay, number 5. Number 5 again is a clear liquid, not very viscous at all. I'm going to waft it. This one has no smell at all. Test it with my pH paper. It's different from the others, Mrs. Sanborn. Not getting too much of a color change on that one, not like the acids and the bases. So this one we might consider to be our control sample. And if you look at it, it's right around in the 7 range, okay? So that would tell us that it's neutral, okay? So that should give you a pretty good idea of what that might be if it's neutral and it has no smell and it's different from the others.
Yes, and it looks like what it is too.
Yeah, it does look like what it is.
Whereas this and this one look the same, but if you
No.
open them, you know. So I'm doing number 6. It's clear and liquid and not viscous at all. It looks very much like the previous one, number 5. When I waft it, it smells like pickles.
Yeah.
It
okay.
smells like pickles.
Or
to me.
salad, maybe.
Yeah, or salad.
Yeah.
Dressing, probably yeah. But okay, so put the lid back on, get out a pH paper.
Oh, you definitely got a reaction there, Mrs. Sanborn.
Yes, it's definitely changed color, and we're back in the oops, wrong end. Let's see. There we go. So this one also is in the 1 to 3 range. It's probably not quite as dark as this one seems now at this point, number 4, but it definitely is in the same range, the 1 to 3. So, and that would be the end of it.
Okay, yeah, a couple of comments we wanted to make though too. You'll notice that all the caps stayed on. This is proper lab technique. And we didn't have little pH papers all over the tray. We kept them right on the top of each sample that it belongs. We want to have good lab technique when you do this. We don't have spills all over the place. The other thing is we used all our safety tools, except for one, Mrs. Sanborn, one that I forgot to mention in the beginning. We used our gloves and our goggles. And what is the most important safety tool that we use in the laboratory?
Our brain.
Yes, using your noggin, not acting like a potato head in the lab, all right? So we used the scientific method today in order to hopefully correctly identify unknown samples. We made observations, we asked questions, we did a laboratory test, a control, we wrote our data down, and now you can work with each other to see if you come up with the correct answers. There is a document on GoFormative that you'll be able to fill out to complete today's exercise, and I hope you enjoyed it. Thank you, Mrs. Sanborn.
Thank you,
Mrs.
Ms.
Shaw, for filming.
Thank you for filming, Ms. Shaw.
No problem.